All right, welcome to the 2021 Virtual Vanderbilt Summer Science Academy. Today is Monday, so that means that we have presenters who have gone the PhD route. And for this week, we are having presenters who are done with their training and actually have a job. And that's exciting. That happens in real life. So today we have well, I would call her a friend now. Katie Hutchinson, who she helped me with the VSSA program back in 2016, I think it was.
Mhm.
So for those of you who are in person, she's the one who made the symposium what it is. She implemented the judging. She had us have students submit their abstracts so we could have oral presentations. She is just a rock star. And now she's got a real job. So let's hear from her. Thank you, Katie, for being here. It's great to have you.
Oh, no, thank you. Thank you. I think you give me too much credit, Beth, but thank you very much. And thanks to all of you for joining and those of you joining on YouTube later. Yeah, so as Beth said, yes, I do consider her a friend as well. It's amazing to see how, just a few years and things have changed and everything. Anyway, yeah, my name is Catherine Hutchinson. I do go by Katie. I'm a PhD, or I have a PhD from Vanderbilt, and I'm currently a principal scientist in the oncology biomarker development group at Genentech, which is, of course, a member of Roche. Which I will explain all that later. That was a lot of words. But essentially, hopefully I can provide you some insights on how I've kind of followed my gut toward my current career in science. So lots of fun photos coming. So Beth was, when she asked me to do this, one of her prompts was there some sort of aha moment that kind of told you, or a switch in your brain that told you that you were going to pursue a career in science? And I can honestly say no. There was just always some sort of gut feeling in the back of my mind that I would someday pursue something in science. And I think a lot of that had to do with my upbringing. My parents were very, I guess, they allowed me to explore lots of things, both inside and out of school. Like, yes, you have to do well in school, and we encourage you to do well in math and science, but we encourage you to explore a lot of other interests as well. So it really could have gone one of two ways for me. I could have had a career in STEM, which obviously I do, or I could have had a career in the arts and arts. So something in music or something in dance. So, I really enjoyed my science and math classes, but I also really enjoyed performing with the pre-professional ballet company that I was a part of. I really enjoyed helping my dad pull apart all his old tractors and cars and trucks. But I also really enjoyed playing the flute and playing the piano and drawing and painting. But at the end of the day, I was kind of a bit of a contrarian and a bit of a nerd before the nerd term was culturally cool, I guess. I hope that's okay to say. And I oscillated among the typical things that a kid growing up was interested in, being an astronaut, being a medical doctor. And when I say I was a contrarian, well, I'm not sure if I said this, but I grew up in a very small rural Indiana town just across the river from Kentucky. And the other girls in my small town just weren't interested in science and math. So of course I had to be interested in science and math, and better than the boys too at science and math. And while the other girls were watching chick flicks and rom-coms and things like that, I was always rushing home from ballet class to watch a recording of Star Trek Next Generation or to watch Star Wars for the millionth time. So again, probably before those things were considered culturally cool. But nonetheless, with the examples that I had in my small hometown, really the only example I had of someone with a career in science were medical doctors. There was no research in my little town, at least none that I was exposed to. There were no environmental science programs or anything like that. So it really was just the medical doctors that I was seeing pursuing something in science and being looked up to. And I think I really admired that, as I think a lot of people do. So in my summers in both high school and college, it's not surprising that I shadowed various physicians, just called them up and said, hey, can I shadow you? I also worked at a pediatric office doing electronic medical record data entry in the early days of EMR. And probably the most fun job I had, and it really was a fun job, was as a night shift patient transporter at the local hospital. So transporting patients from intake to surgery or from their room to a CAT scan, things like that. So as you can see, everything I was working toward was carrying me toward a career as a medical doctor. So when it came time to go to college, I chose to go to Centre College. And there were really two main reasons for that. One was that I knew no one at Centre. Centre is near Lexington, Kentucky, for those of you that don't know. And I did that on purpose. I just wanted to experience new people, new ways of thinking, things like that. And their medical school prep program at the time had a 98% success rate in getting their students in on the first try to apply to medical school. I hope that made sense. So I was like, this is the place for me. I have to go here. So originally I was a chemistry major, but the first couple of years really kicked my butt, if I'm honest. I mean, everyone at Centre was on some other level. And what I really came to realize was that I didn't really know how to study. I could memorize facts and regurgitate facts, which you could argue is useful for medical school. But it just wasn't going to fly at a small liberal arts college to just memorize and regurgitate facts. So I really had to relearn how to study and how to think critically, which was a big morale suck for me. But on the bright side, I started taking classes that I had only brushed upon in college, or excuse me, in high school, things like virology, molecular biology, biochemistry. So I started taking actual courses in those, and those were in the biochem and molecular biology department, or the B&B department. And I don't know what it was, but everything just kind of clicked. I was really excited. Perhaps it was the novelty, but I was really excited to learn and relearn how to learn so that I could learn more in those classes. So I ultimately, I should back up and say that I think a huge influence on this was my now husband, Jordan, who's also a scientist. He and I met in the B&B department, and he really encouraged me to switch because he saw how much I was not loving all the chemistry courses. And so he encouraged me to switch, and I think it was a good decision for me. Yeah. So I guess what else I'll say about the B&B classes is that what I also really liked about those classes is that I could see a clear link between the content in those courses and how that work applied to human health and healthcare, which is something I was still interested in, even if it meant not being a medical doctor. So I did apply to and was accepted to some grad schools, but obviously all my summers to this point had been spent preparing for medical school. So I had basically no laboratory experience outside of our class laboratories. And I really did want to go to Vanderbilt. Part of it, again, was my husband. He was pretty set on, or my then boyfriend, but he was pretty set on going to Vanderbilt. And I also felt that Vanderbilt, compared to the other schools that I did get offers from, just was on another level and would set me up for even better future success. So I went with my gut again, and I declined those other grad school offers. And instead I applied to a bunch of labs to be a laboratory technician and assistant so that I could gain some experience and hopefully reapply and get accepted to Vanderbilt the next year. That worked out. My gut is always a good place to start. And I was accepted as a research technician or research assistant in the laboratory of William Powell. And this was in the former cancer biology department. I know that that has been absorbed into one of the other departments now. So William had just come from Memorial Sloan Kettering. He is an MD PhD. So at the time he was seeing patients as well as running and starting up his own lab here at Vanderbilt. But I was partnered with one of his senior graduate students that came with him from New York. And there I learned some really practical experimental techniques to better understand cell signaling, cell biology. And I also was able to get my hands on some what were then at the time emerging genomic sequencing technologies. So you may have heard about next generation sequencing. And it was very new when I was just starting out. So when I reapplied to Vanderbilt's program in 2010, I was accepted. Yay, my plan worked out. And the other plus was that my husband was also accepted in the 2010 year as well. So we were able to start together and have moral support. And I have to say, you know, taking that year off instead of going straight to one of another grad schools that I had been accepted was a really, really good decision. It really gave me a leg up to focus on an area of research and understand it well and understand the techniques well. Obviously everyone's different, but it really gave me a leg up. And I felt like the core courses in the IGP were a breeze for me. So that's definitely a switch from the last time I entered like a college setting. Everything was, you know, not easy peasy, but very doable. And so after the IGP rotations, the integrated graduate program rotations, I did rejoin Dr. Powell's lab for my own graduate work. And for my graduate work, we essentially were able to access tumor tissue samples from patients with cancer. So using the sequencing data from those patients' tumor samples, and these patients typically had lung cancer or pancreatic cancer or skin cancer. And so we would take the sequence data from those and identify mutations that were associated with resistance to the treatment that those patients were receiving as part of their cancer care. And then what I would do is take those mutations and model them in cell line models and mouse models to understand the mechanism behind why those mutations do what they do. And then hopefully help identify potential alternative treatments or combination treatments in the lab to then maybe someday bring that back to the clinic. That was the ultimate vision. So that was really exciting. And as you can see, we had a lot of fun in the POW lab. And so for my postdoc, we kind of had, when I was done with my PhD work in 2014, 2015, excuse me, my husband was not yet done with his PhD. And so we had a real two bodies in science problem. Which is a real thing. So I was ready to move on. He was not done with his graduate work. It's not like he can take the lab with him, right? So I decided to remain at Vanderbilt for my postdoctoral work. And I moved to the laboratory of Carlos Arteaga. And in Carlos's lab, I was able to apply a lot of the work that I had done as a technician and as a graduate student in lung cancer, pancreatic cancer, and melanoma to patients with breast cancer. So again, modeling molecular mechanisms of resistance to some new targeted therapies that patients with a specific type of breast cancer were being treated with in the clinic. So that was really exciting. So again, we had this two bodies in science problem. It seems to be just the ultimate theme of my life. What that means is like the next steps and next location and timing were unknown for us. And so I used a lot of my time as a postdoc and part of it with Beth and the VSSA to explore alternatives because I didn't know where we would be. I didn't know what would be available to me wherever it was that we would move, but we knew that we would be moving out of Nashville, probably to the East Coast or West Coast. So I prepped for both industry and non-industry positions. I ended up getting my college teaching certificate while at Vanderbilt. And again, I also helped Beth with the VSSA, which was, you know, also helping educate newer scientists was something, and still is something that I am interested in. But ultimately, we lucked out on timing. About the same time that I received a couple offers from for industry positions in both Boston and San Francisco, Jordan, my husband, who was at this time finally my husband, he received a postdoctoral offer with Roger Kornberg at Stanford University. So obviously our gut said, let's go to San Francisco. And so that's where we are now. And again, at Genentech, I'm a principal scientist in oncology biomarker development. And while I am no longer at the bench, I would say that a lot of my bench work and a lot of my training up to this point, and a lot of the career advice that I got from folks at Vanderbilt really set me up for success for this position. So again, instead of being at the laboratory bench and doing experiments, I actually lead teams. So I lead teams that are responsible for drug development from the biomarker side. And so I currently support molecules in our Roche Genentech pipeline for patients with breast, lung, and pediatric cancers. And something that I do still continue to do that has carried over from graduate school and postdoctoral times is that I continue to publish in scientific journals, as well as attend and present at domestic and international conferences, which is definitely a perk of working for the company. And something that no one has ever trained for, at least not in any sort of grad school setting that I've heard of, but I think is really, really cool and one of my favorite things to do is preparing for regulatory interactions with the FDA and the EMA. So I prepare the biomarker science portion of our clinical trial proposals to the FDA and the EMA, so the EMA being the European version of the FDA. I present that to the FDA and the EMA and have back and forths with them and have to, make our case with them. And so that's something, obviously arguing points and science and facts is what we are trained for, but I just never really thought I'd be having regular meetings every 6 months with the FDA. And I just think it's really cool. So yeah, that's what I do now. So it's all really kind of come full circle for me. In grad school, my job was to identify resistance mutations to targeted treatments for patients with lung, pancreatic, and skin cancer. I transferred that to my postdoctoral work to identify resistance mutations for patients with breast cancer. And now I'm applying all that knowledge to make sure that we design smart clinical trials, so identifying the right patients for our treatments. And all of this is contributing to PHC, personalized healthcare, personalized cancer care. So again, matching the right patient with the right tumor to the right treatment at the right time. One of the problems that we still have, though, is that a lot of the oncology biomarkers that we've identified, so the mutations and other biological changes that are happening in tumor cells that are predictive of response or resistance to treatment, are super, super rare. So in a population of patients with cancer, these can be less than 5% and even less than 1%. So how do you design a clinical trial and how do you use the existing model, which I'm exaggerating here, the traditional clinical trial design, testing one drug against one disease or against one biomarker? How can we make clinical trials feasible when it's really hard to find these patients? And so one solution is to design a new type of trial, merging the basket and umbrella trial features. So again, in a traditional clinical trial design, a little bit of an exaggeration, you're testing one drug or one combination of drugs against one disease or one biomarker. In basket trials, you can have many different types of cancers, but they all have to have the same biomarker against the drug you're testing. So that's one type of novel clinical trial. In an umbrella trial, you have kind of the opposite, one type of cancer in which you're looking for multiple biomarkers that can then be matched to multiple different drugs. But what we're starting to do now is make baskets of umbrellas or umbrellas of baskets and merge the two. So we can have a multi-tumor, multi-biomarker, multi-drug trial. And it really saves us a lot of time, resources, and it's really what's best for the patients. And I have to say, this is a concept that Dr. Powell, my graduate advisor, and I were talking about however many years ago it was. He's like, I'd really love to see this happen. And now it's happening. So everything is just kind of coming full circle, and just really rewarding to see how drug companies are following the science. And the health authorities too are understanding the importance of needing to find the right patients for the right drug at the right time, as well as the need for efficiency. So that's my spiel on coming full circle. So I guess I'm kind of at the end here. I do have a few key takeaways, which are shouldn't be surprising to anyone, but it's okay to change plans. It is okay to not know what you're doing all the time, because I certainly do not and did not just kind of follow where my gut told me to go. Something that I wish I had done more of is ask questions and ask more questions. Even if it seems silly, just ask the question, because then you'll be saved from suffering in silence, which is what I tend to do, because I like to absorb and think through things and then ask my questions. But just start asking questions if you can force yourself to do this. Utilize your network. If you've met someone or you worked well with someone, utilize them to help you find that next step, or find a mentor, or find a new job. You never know what can happen. So use your network. One practical thing that I wish I had done that probably wasn't available to me at the time at Vanderbilt is to learn large-scale data analysis or bioinformatics. I can't tell you how many job applications I see or job requisitions I see where one of the number one requests is that you know how to do large data sequence analysis. So if you can take a course, maybe you don't have to become a computer science expert or an informatics expert, but just at least have an understanding of how all that works will be a huge boost to your resume for grad school, postdoc, beyond postdoc. And obviously find time away from the lab. I think this last year has really shown all of us, especially this last year, that being stuck at our bench, being stuck in our books, being stuck at our jobs 100% of the time with like no real separation is just not good for our physical or mental health. So and this is true before the pandemic too, but I think especially this pandemic has really allowed a lot of us to reflect on how important the time away from those things is as much as the time with those things is. And so you can see we do take advantage of the outdoors out here in California and with our awesome dog. But find time away from the lab because it's really refreshing, even if it's just a day or an afternoon or a quick, walk around the block or something, because it will change your attitude toward the way you approach your work. And maybe some new ideas will pop into your head. And yeah, it's just always a good idea. And the end, follow your gut. That's all I have to say. Your gut is always a good place to start. So that's it for me. I hope that's been useful. Beth, I'll turn it back to you.
That's wonderful. Thank you so much, Katie. I think sometimes when you hear about somebody's story, it might seem like an intentional linear path and everybody knows exactly what they're doing. And I just don't think that's true.
No.
So thank you so much. I'm going to open it up to questions, but while anybody types their questions or raises their hand, let me ask you, when you were making the transition from thinking about med school to then grad school, was there, well, maybe what I want to ask is, how do you feel looking back about that? Was that the right move? Do you ever think about that as something like, would you consider med school now if you could do it again? Talk a little bit about that transition.
Yeah, no, that's a great question. I think at the time, it was a little scary, because I had just always thought I would be a medical doctor, at least in high school, for sure. And before that, astronaut, that's kind of funny. But yeah, no, I think at the time when I did that transition from, med school prep to prepping for research-based career, it was a little scary. But I do think it was the right decision for me because I really enjoyed the autonomy of research, the research track. And yeah, there will always be that kind of unknown, what if I had been able to do it, or what if I went back to do med school or something like that. But I'm actually, I'm very happy with how things have turned out. And yeah, no regrets really, but it was a little scary at the time, not gonna lie. Is there a question in the chat too? Oh, it just said thank you. You're welcome. Okay.
Does anybody else have a question? I might have one more. So, because I need this advice to take a moment away and take a break sometimes. That's okay.
Yeah.
How do you manage that? Do you schedule breaks or do you do it on a whim? Talk a little bit about how you make yourself do that.
Well, I will say it's easier than it used to be in grad school. There's just this unspoken pressure, just like you have to be at the bench all the time, even on Saturday and Sunday. And maybe that's true for some people. I just always felt that pressure, though nobody ever told me that. But now, yeah, I will say that my work schedule now is conducive to just like piecing out at 2:00 in the afternoon for a few hours and then coming back. Because a lot of my work is global. My mornings start at 7 and end at 2, basically. So that's, a solid 7 hours already. Of meetings, documents. So at 2:00, I do try to take like a break, take the dog for a walk, go to a hike real quick, and then I'll just come back a little refreshed later. And I do schedule it out. Sometimes that doesn't work, and I don't do this every day because it's not sustainable, but yeah, just schedule breaks in your day. And our company has been really good too about like encouraging us to block out time in our calendar to step away from the computer because it is fatiguing. And then, for some of the longer trips, my Fridays are quite light on meetings. I think Europe is asleep and everyone is tired. So Fridays I can usually hop away by noon if I needed to go to like, the mountains or something to get away. But yeah, just kind of schedule it and be diligent about your time and be okay saying no, which is something I'm still learning.
Teach me when you figure it out.
Okay.
Thank you so much, Katie. It was awesome to see you.
And me too.
Thank you so much for sharing your story. I really appreciate that.
You're welcome.
And I know you got lots of meetings, so we'll let you go, but thanks for being here.
You're very welcome. Yeah. If any of you have any questions, I'm on LinkedIn, so I'm available. Yeah. Thank you so much, Beth.
Thank you. Good to see you.
Bye.
Check out our website to find recordings of all of our videos. We'll get them posted about a week after each of the presentations. We can't wait to see you.